In this video, we're going to talk about the polymerase chain reaction, or PCR. Now, this technique is very useful because it allows us to amplify a single DNA molecule to produce billions of molecules of DNA of the same sequence. Now, we can apply the PCR technique if the starting sample contains only a small or only a single molecule of DNA. And that might be, for example, from a saliva sample or a blood sample from a crime scene. So we might want to amplify that DNA before we can start to analyze it. Another example of where PCR is useful is in paternity testing, or testing if two people are closely related. So we might take a swab of cheek cells from the inside of the cheek. We might put that in a little bit of fluid. We then isolate the DNA from the cells, and we can amplify certain parts of the genome and compare between two different people. So we know within our somatic or body cells, each nucleus has 46 chromosomes. So there's 23 different types of chromosome, but there's 2 of each because we're diploid, so 46 chromosomes. Now, within the genome, which is the entire sequence of bases for all the chromosomes inside the nucleus, we find sequences called introns. Now, introns are non-coding, so non-coding parts of the DNA. So we know there's a sequence of bases, but they just don't code for any proteins or any part of a protein. So we can get introns within a gene or in between genes. Now, in the genome, we have intron sequences that are called hypervariable. So hypervariable regions. And there are many of these hypervariable regions within the genome. So if we start with hypervariable region 1, within this hypervariable region that can be different between different people, there are what we call short tandem repeats. So this one is AGAT. And we can see here this AGAT, the short tandem repeat, is repeated 7 times. So in this DNA from this person, in hypervariable region 1, there's an STR or short tandem repeat of AGAT, and it's repeated 7 times. In a different part of the genome of that person, there's going to be a second hypervariable region, so we'll call that number 2. And in this second hypervariable region, there's a short tandem repeat made of GATA, so GATA. Now this GATA short tandem repeat in this area is repeated 6, 7, 8 times. Now what we can do is we can amplify these hypervariable regions using PCR from a single person. Now if we did that for a second person, we can then start to compare them and look at how many repeats we find within each hypervariable region. So let's have a look at the second individual, and this is their DNA. So if we look at hypervariable region 1 from the second individual, we know it's AGAT, it's a short tandem repeat. Here we've got 7 of these AGATs repeated. So it's actually the same as the first individual. But if we look at the second hypervariable region from the second person, we know it's GATA. This time, if we count how many GATA repeats we have, we'll actually find there's 13 of them. So in this second person, we know that the second hypervariable region is bigger compared to the first person. So this band would be a bigger band of DNA that gets amplified. So this can apply, for example, if we looked at a crime scene. If we took a sample of blood or saliva where we can extract DNA, and we amplified hypervariable region 1 and then hypervariable region 2, we can compare these sequences to maybe a suspect's DNA. So we amplify using PCR the same regions. Now, the first hypervariable region would actually give us the same DNA fragment. So that's exactly the same. But when we do this PCR for the second hypervariable region, we can see it's actually a lot bigger in the second individual. So we can see it goes all the way down here. So there's 13 STR repeats, which is obviously different from the DNA from the crime scene. So we could say here that the suspect's DNA does not match the crime scene DNA, and therefore you might exclude this suspect from the inquiry because their DNA does not match that of the crime scene. And we can use this technique for paternity testing or looking at how close certain people are related to each other. Now, more closely related people will tend to have these hypervariable regions that are more similar in size. So we can look at a number of different hypervariable regions and the number of STRs, or short tandem repeats, there are within each hypervariable region. We can then start to build up what we call a unique genetic fingerprint for a person. Now, if we compare these analyses between person 1 and person 2, we can say if they're closely related or not. And we use the PCR technique to amplify the DNA to start with. We then run the DNA on an electrophoresis gel and we look at the size of the bands. Now, we know in PCR that we're going to start off with a single DNA molecule. Now, at the end of the first cycle, we basically double the number of DNA molecules that we have. So we'd end up with 2 DNA molecules at the end of cycle 1. Now, these 2 DNA molecules are exactly the same sequence and exactly the same size as the original DNA molecule that we started with. Now, the second cycle, we're going to take 2 DNA molecules, and at the end of the second cycle, we'll have 4 DNA molecules. And again, these will be exactly the same size and exactly the same sequence. Now, we know DNA replicates in what's called a semi-conservative manner. So what happens is each DNA molecule during the cycle will separate. It's not DNA helicase here that separates, unwinds the DNA and separates it. The DNA is separated through the high 95 degrees C temperature. Now, each DNA strand acts as a template strand for the synthesis of a brand new strand. So each DNA molecule consists of the original DNA strand and a brand new, newly made strand. So if we take these 4 DNA molecules and we have cycle number 3, we end up obviously with 8 DNA molecules at the end of the 3rd cycle. Now, these again will be exactly the same size and exactly the same sequence as the original DNA molecule that we started with over on the left. So you can see they're all exactly the same size and sequence compared to the original DNA molecule. So as a general rule, we can use this equation that we've got in the red box. So 2 to the power n, where n is the number of cycles of PCR. So 3 cycles of PCR would give us 2 to the power 3 is 8 DNA molecules. So if we had 5 cycles, then obviously that's going to be 2 to the power 5. If we had 15, 2 to the power 15, and 38 cycles would give us 2.75 times 10 to the power 11 DNA molecules. So we started with a single DNA molecule, and we've ended up with billions of the same sequence and same size DNA molecule. Now, in reality, we actually don't get this number at the end of 38 cycles, and that's because normally the DNA nucleotides do become limiting in the reaction mixture, because we know they get used up to build the new strands of DNA. When we talk about PCR, we need to talk about the different components in the reaction mixture. So we're going to have a specialized machine called a thermocycler, and in that machine we can switch between 3 different temperatures: 95 degrees C down to 55 and back up to 72. Now that would be 1 cycle, and that gets repeated 38 to 40 times. So in our PCR reaction mix, we start with the DNA sample. Now this sample is obviously double-stranded DNA. It's taken from maybe a blood sample or a saliva sample, or even from within a cell if we break open the cells. Now, the next components we add in are what we call primers, and we've got 2 types of primer. Now, these are quite short 10 to 20 base single strands of DNA. So they've only got between 10 and 20 nucleotides in these single-stranded pieces of DNA. Now we need primers because these anneal to the single-stranded DNA, and from the primer, the new strand of DNA can then be built, because DNA can only be built off an existing structure. So we'll see that in a minute. Now the next component in the PCR mix are the DNA nucleotides. So we know a DNA nucleotide each is made from 3 components: the nitrogenous base, the deoxyribose sugar, and a phosphate group. So nucleotides are the subunits of all nucleic acids, and DNA nucleotides are the subunits of a single polynucleotide strand. So in excess, we've got 4 different types of DNA nucleotides. One of them will have cytosine as the nitrogenous base. One type of DNA nucleotide will have guanine as the base. One will have thymine, and one will have adenine as the nitrogenous base. Now the next component is the kind of special ingredient, and this is called thermostable Taq DNA polymerase. Now this DNA polymerase is isolated from what we call a thermophilic bacteria, and it's highly resistant to high temperatures. So normally an enzyme, for example the regular DNA polymerase from within human cells, would denature above, or start to denature above 37 degrees C. And that's because the enzymes vibrate more, and that breaks the hydrogen bonds between the variable groups within the tertiary protein, and it changes the shape of the globular tertiary protein and changes the shape of the active site. So you don't get any more successful collisions, and you don't get any ES complexes being formed. But this thermostable DNA polymerase, it does not denature even at very high temperatures of 95 degrees C, which we use within the thermocycler. So Taq DNA polymerase does not denature at the very high temperatures, and that's very useful for this reaction. Now we're going to have a mix called a buffer into which all these ingredients go, and we then put that mixture into a PCR tube, and we place the PCR tube in the thermocycler machine, and we basically click go for 38 cycles. So the actual process itself then, well, we know the DNA that we start with, the sample DNA, is double-stranded. Now the first temperature is called is 95 degrees C, and that allows what we call denaturing to take place. So that separates the 2 DNA strands because the hydrogen bonds break between the complementary base pairs, so it separates our DNA strands. Now both DNA strands are going to act as templates, so they both act as templates for new DNA strands. Now 55 degrees C is the next temperature, so we get a big drop in temperature from 95 to 55 degrees C. This allows our primers, single-stranded short DNA molecules, to anneal or bind with a specific sequence. So the bases of the primers, complementary base pair with a certain sequence of DNA. So we can see that here. Now what we then do is we increase the temperature in the machine to 72 degrees C. Now because there's DNA nucleotides within the mixture, the DNA nucleotides align next to the exposed bases. So they naturally align next to the exposed base because of the complementary bases that we know interact. So these DNA nucleotides align next to the complementary base, and the Taq DNA polymerase at 72 degrees C allows extension. So the DNA nucleotides that have aligned next to the exposed bases on the template strands are joined together by the DNA polymerase, and it forms the sugar phosphate backbone of the brand new strand of DNA. So what we end up with after a single cycle is 2 molecules of double-stranded DNA. Okay, so we've got 2 molecules of double-stranded DNA. Now we know each molecule of DNA by the semi-conservative method of DNA replication consists of an original DNA strand from the original DNA molecule and a brand new, newly made strand. So each molecule, if we take this one here on the bottom of the page, we can see there's one original strand, and against that has been made a brand new strand of DNA, and that's called the semi-conservative method of replication. So we can start to look at this in a little bit more detail. So we know we separate the two strands at 95 degrees C. It breaks the hydrogen bonds and we get two template strands of DNA. Now, DNA is always synthesized in a 5' to 3' direction. And we know double-stranded DNA is what we call anti-parallel. So this DNA strand at the top, this single strand of DNA, it runs 5' to 3'. So the new strand that's going to be built against this template strand, we know is built 5' to 3'. So we're going to see in a minute, the brand new strand is going to go in this direction, because DNA is always built 5' to 3'. Now, on the other template strand of DNA, we can see the 5' end is here and the 3' end is here. So this single strand of DNA runs in that direction. So when we build the new strand against it, the second strand is going to be in this direction, towards the 3' end. So we've got our separated DNA. We've got two template strands, single-stranded. We now add our primers that we've specially designed for the PCR reaction. So primer number 1 is at the top, and primer number 2 is just beneath it in pink. So we know these are going to form hydrogen bonds with their complementary bases. Now this occurs at about 55 degrees C, and we call that annealing. So primer number 1 anneals there, and primer number 2 anneals here. So you can actually see the complementary bases that hydrogen bond between the template strand and primer. So this one here would be primer number 2, and the one over on the left we can see there is primer number 1. So this is called annealing. So the temperatures drop from 94 or 95 degrees C right down to 55. This allows the primers to anneal to their specific complementary sequence. And then the next step is we're going to increase the temperature up to 72 degrees C. So the next step is called extension, and this occurs at 70 to 72 degrees C. Now, we know the mix contains DNA nucleotides. Now, we know these DNA nucleotides will align and sit next to the exposed base on the template strand. So the DNA nucleotides do not link together until DNA polymerase has catalyzed that reaction. So they'll just sit next to the exposed base on the template strand. We know DNA polymerase will work off an existing structure, and that is the primer. So for example, this primer we said was number 2. Now that starts at 5', and this G nucleotide at the end would be the 3' end of the primer. So DNA polymerase will start to link together the DNA nucleotides in the new DNA strand from the thymine going to the left here towards the 3' end. So down here, where we've got the dotted lines, we would have DNA nucleotides that are complementary to the template strand. DNA polymerase thermostable would move from 5' to 3'. It would link together all these DNA nucleotides, form the sugar phosphate backbone, and we'd end up with the 3' end of the new strand on the left hand side. And it's the same for the other template strand of DNA. We know our primer number 1 has annealed. We know the primer is 5', so the G here would be 3'. DNA polymerase will build off an existing structure, so this adenine here will link to the G next to it, to the T next to it. So it's built 5' to 3'. So the DNA polymerase moves this way as well. Now, these DNA nucleotides that sit next to their complementary base will be linked together to a new sugar phosphate backbone, and this will be the 3' end of the new strand against the second template strand. So this is called the extension step. So a brand new polynucleotide strand is built against both original template strands in a 5' to 3' direction. Now, if we looked at this in a bit more detail, we could actually draw a graph of the number of DNA molecules being produced as the number of cycles increases. So, for example, on the x-axis on the bottom, if we plotted the number of PCR cycles going up to 40, and on the y-axis we had the number of DNA molecules, what we could see is an exponential increase in the number of DNA molecules at around about 15 to 25 cycles. Now, we can see the number of DNA molecules when there's a low number of cycles is quite low, because we go from 1 to 2 to 4 to 8 to 16, etc. So it's not going to increase massively during the first 10 cycles or so. But when we get to a bigger number and we start to double the number of DNA molecules each cycle, we get this exponential increase. But what we'll see, as the number of cycles progresses towards 40 cycles, the number of DNA molecules starts to plateau. So we know something must be limiting at this point in the reaction. And we know DNA nucleotides that we add to the mix at the start of the reaction get used up. So it's actually the DNA nucleotides, as they get used up, they start to become limiting. So the number of DNA molecules starts to plateau at the very end of the 40 cycles or so. So we can have a look at a couple of questions then. During step number 2, which we know is the annealing step, short DNA primers need to be added in order for the DNA synthesis to proceed. Well, they're actually already added to the mix. They just need to be able to bind to the template DNA. Explain what is meant by a primer for 2 marks. Well, we know there's 2 types of primer, and there's many of them within the mix itself. We know each primer has a specific sequence, and it's going to be between 10 to 20 nucleotides long. We also know they're going to be single-stranded. And what you could also add is that we know they hydrogen bond, i.e. anneal, to the complementary template strand of DNA. Now, without the primer, the Taq DNA polymerase cannot synthesize a new strand. That's because any DNA polymerase needs a primer to build off to be able to synthesize a new strand of DNA. So new strand. Now, which enzyme is required at step number 3? Well, step number 3 is the elongation phase, or the synthesis phase. So we know it's Taq DNA polymerase, and we know it's a specialized thermostable, DNA polymerase, because one of the temperatures is 95 degrees C, which would otherwise denature and inactivate most DNA polymerases, because it changes the shape of the active site, and we don't get any successful collisions between the active site and the substrate, so we don't get any ES complexes, and therefore the new strand would not be normally made. So we can write that down. The thermostable DNA Taq polymerase is very, very stable at very high temperatures. We know it's not denatured at these high temperatures. It's very important in PCR because we know we cycle the temperatures 95 to 55 to 72, and 95 would normally denature most of the enzymes that we find elsewhere. Now, we can actually look at this PCR reaction in a little tiny bit more detail. So at the top here, we've got double-stranded sample DNA, and we want to amplify a certain sequence. So it's going to be between this point on the left and this point on the right-hand side. Now, this can actually be quite useful, because imagine we had human DNA and we wanted to amplify maybe the insulin gene, for example. Now, because we know the sequence of the insulin gene, we can actually design primers that we know can anneal to certain parts of the DNA, probably just either side of the gene. So we can actually amplify very useful sequences of DNA. Now, if we amplified the insulin gene, we could then put that DNA into a bacterial plasmid to form recombinant DNA. And we could then put the recombinant plasmid into bacterium and allow the bacteria to divide. Now, each bacterium that had the recombinant plasmid would then start to express the human protein, the human insulin protein, from the human insulin gene that's been inserted into the plasmid. So we know 95 degrees C is going to separate the two DNA strands, polynucleotide strands. It breaks the hydrogen bonds, and each DNA strand is now a template. We know when we increase the temperature to 55 degrees C, the two primers are going to bind to their corresponding complementary sequence. So here, if we look at the DNA strand on the bottom, it's 5' to 3'. So we know the primer here on the left goes 5' to 3' in the opposite direction. Now, if we took this scenario and we elongated from the primers, this is what we'd see in the very first cycle. So we know these are our two primers in red, and we know there's DNA nucleotides in the mix, and we know Taq DNA polymerase is going to build the strand in blue from the primer in a 5' to 3' direction. So going this way. Now, this would be the end of the first cycle, actually. Now, if we actually denatured these double-stranded DNA molecules and we then did a second cycle, we'd start to amplify these sequences. So imagine the sequences, the colored sequences, have been separated from the original template strand. So at the bottom here, these two single-stranded pieces of DNA are actually the colored strands from above. So we haven't drawn in the black strands from above, just to make it simple. Now, the second cycle then, well, if we added, if we changed the temperature to 55 degrees C, so we know we've denatured at 95 degrees C, the primers would anneal. If we then allowed 72 degrees C temperature to be reached, we know DNA polymerase would synthesize the new strand against the template strand, but this time, when the new strand is being formed, 5' to 3', it can only go up to this position because there's nothing else beyond this point. There's no other template to build against after this point. And it's the same for the double-stranded DNA at the bottom. We build in a 5' to 3' direction. So once we get to this point, there's nothing else to build. So we're now going to take these two double-stranded pieces of DNA. We go into the third cycle. So we can see that next. We separate the double-stranded DNA at 95 degrees C. We drop the temperature to 55. That anneals the primers. We then synthesize and elongate at 72 degrees C. And we can now see, this is the third cycle, that we have double-stranded DNA of a specific length and a specific sequence. So here we have two DNA molecules, both the same length, both the same sequence. Now, if we denatured both of these DNA molecules again, what we'd find is that the next cycle, we'd get brand new DNA molecules. So we'd end up with 4 DNA molecules in the next cycle that are exactly the same length and exactly the same sequence. So this goes up, as we said, 38 to 40 cycles. Now, we also need to know what could be a negative of PCR. So it's a very precise technique. So imagine you had a DNA sample and you wanted to amplify a sequence. Well, if that DNA sample was actually contaminated with a different DNA sample, maybe from a different person, then within the same mix, you could actually end up amplifying 2 different sequences because you've got 2 DNA samples. Now, the second issue with PCR is that DNA polymerase because the Taq thermostable DNA polymerase is taken from a bacterium, the bacterial DNA polymerases do not check which nucleotide has been added to the brand new strand of DNA. So Taq DNA polymerase does not proofread the new strand of DNA to make sure it's the correct nucleotide that's been added. Now, the DNA polymerase from our cells, from our nuclei, that does proofread the DNA. So when we make brand new molecules of DNA during DNA replication, the DNA polymerase actually proofreads the brand new strand of DNA to make sure it's free of any errors. But this bacterial polymerase does not do that. So PCR can introduce occasionally some errors. Now, we also know PCR is quite susceptible to inhibitors. So certain chemicals from the environment can actually inhibit by interacting with the DNA or interacting with the enzyme, the process of DNA synthesis. So, for example, we get phenolic compounds from certain plant materials, and they can inhibit the Taq DNA polymerase. Quite often, certain chemicals in things like the dyes used to dye genes can also inhibit DNA polymerase. Now, another one is that the sample of DNA you have, whatever the sequence you want to amplify is, it has to be of a certain length. So you can't amplify huge, big sequences of DNA. We can only amplify certain sequences. Now, we also know after a certain number of cycles of PCR, some of the reagents will become limiting. So, for example, the DNA nucleotides, because we only add a certain number of them at the start of the process, these get used up, and so they can become limiting towards the end of the process. Now, the optimal length to amplify a sequence of DNA is anywhere between 1 to 3,000 bases. Anything bigger than 1 to 3,000 bases is unlikely to amplify correctly because there's going to be more errors introduced into the amplified molecules of DNA.